chromosome 10 is extraordinary. We all know what it's like if we're forced out of bed way before 5 o'clock in the morning. It takes us a long time to wake up. We all have sort of a day within a biological clock, also called a circadian clock. And what this does, and it really can't be overemphasized, is regulate almost every aspect of our physiology and behavior, fine-tuning physiology and behavior to the varying demands of activity and rest, cranking up hormones and metabolism in anticipation of activity and turning it down again in anticipation of sleep. So the sleep-wake cycle, changes in blood pressure, changes in temperature, still show a near 24-hour oscillation, but it wouldn't be exactly 24 hours. And so what needs to happen every day is that this slightly drifting clock needs to be adjusted to environmental time. And the most important signal that the clock needs is exposure to the light-dark cycle. And that light-dark is detected in mammals, such as ourselves, by light-detecting cells within the eye. It's critically important that the body clock is adjusted every day by the light-dark cycle. And if you didn't get that daily adjustment, then internal time would be abnormally synchronized with the external world, and perhaps the best example of that is jet lag. So we knew that these photoreceptors were in the eye, because if you prevented light getting to the eye, then the body clock no longer responded to light. So there's something in the eye. The big problem is that it was very difficult to understand how the classical visual cells of the eye could be detecting overall brightness within the environment. For vision to work, the visual cells grab light, they send that information off into the brain, and then essentially they stop working. So you have a series of sharp images. And it was very difficult to understand how that image detecting system could also get an appreciation of the overall amount of brightness and hence time of day. So there was a big puzzle. We started using mice in which the rods and cones had largely degenerated as a result of inherited retinal diseases. So these mice had lost most of their rods and cones, they were visually blind, and we wanted to see whether they could still regulate their body clocks. And what was absolutely extraordinary is that these visually blind mice were able to regulate their body clock to the light-dark cycle perfectly normally. The mouse would get up, run in its running wheel, and then go to bed at the same time, day after day after day. And so we thought, my goodness, there's something really interesting going on here. There had to be another class of light-sensitive cell within the eye. And the big question, of course, is, well, if it's not the rods and cones, then which of the cells of the inner retina could it possibly be? Eventually, the light-sensitive molecule was found, and it was called Melanopsin, or OPN4, and that gene is located on chromosome 10. I think knowledge about this new system is really very important. If you think about it, we know that there's about 40 million people worldwide who have no sense of visual sensitivity at all. Now, understanding that there's another light sensor within the eye could hugely improve the quality of life and the sense of well-being of those individuals. And at the moment, very little is discussed about those cells. So the fact that melanopsin is turning up in chromosome 10 makes chromosome 10 my favourite chromosome.